My name's Andrew Cresswell from the University of Queensland and past president of the International Society of Biomechanics, and it's my honour and pleasure to be able to introduce the Muybridge winner this year. For those of you who don't know, Edward Muybridge was born in 1830 and died in 1904 and was an English-American photographer, especially known for his pioneering work in photographic studies of motion. Today, Muybridge is known for his pioneering work on animal and human locomotion, which used multiple cameras to capture motion in stop-motion photographs. In the 1880s, he entered a very productive period at the University of Pennsylvania, producing over 100,000 images of animals and humans in motion, capturing what the human eye could not distinguish as separate movements. The Muybridge Award, often known as the Muybridge Medal, is the most prestigious award of the International Society of Biomechanics, and is awarded every 2 years for career achievements in biomechanics, and is sponsored by one of our longtime sponsors, Vicon. Candidates for the award are canvassed, and a committee chaired by the ISB past president, myself, selects the winner based on the impact and the quality of their work, taking into account the diversity of research areas covered in biomechanics. It's with that in mind that I'm pleased to be able to present this year's winner of the Muybridge Medal, Professor Ralph Müller, who is head of the Laboratory for Bone Biomechanics at the Institute of Biomechanics at ETH Zurich. In the early part of his career, Professor Müller worked in Boston, where he had a tenure-track position as an assistant professor of orthopedic surgery at Harvard Medical School, and was associate director of the Orthopedic Biomechanics Laboratory. On return to Switzerland in 2000, he was appointed as professor at the Department of Mechanical and Process Engineering at ETH, and in 2012, he was one of the founding members of the Department of Health Sciences and Technology at ETH. The research he has completed and is currently pursuing employs state-of-the-art biomechanical testing and simulation techniques, as well as novel bioimaging and visualization strategies for musculoskeletal tissues. Dr. Müller has an impressive track record, an author of over 320 research papers, 230 proceedings, 80 book chapters, 2 books, 646 peer-reviewed abstracts. His work cited over 31,000 times and has a current H index of 91. He's former president of the European Society of Biomechanics, the Swiss Society for Biomedical Engineering, and currently serves on the board of directors for the International Society of Bone Morphometry and the International Federation of Musculoskeletal Research Societies. He's received numerous awards, including most recently the Mike Horton Award for the European Calcified Tissue Society, the Huskus Medal for Biomechanics from the European Society of Biomechanics, and now, today, the Muybridge Award from the International Society of Biomechanics. I would like now to invite Professor Müller to the stage to receive the Muybridge Medal and give this year's Muybridge Lecture.
Well, good afternoon, everybody. Thank you very much for this nice introduction, and of course, thank you very much for being able to receive this medal. It's a real honor for me and a pleasure. I'm delighted when I heard that, and especially because, you know, this is 20 years after I received the Promising Young Scientist Awards in the same place, 1999, in Calgary, and this is something quite special. So, you know, for everybody out there that has the potential also maybe to go to some of these younger candidate awards, you can see sometimes from promising you can also deliver maybe every now and then and become a more senior recipient of the award. So today, what I'd like to do with you is to take you on a journey through bone, and that will be taken kind of literally to some extent, from mechanics to mechanomics. And I will go a little bit into that, what I mean with it, of course, during my talk. So I have a little prelude to make, and after being with my wife for 35 years, I can attest to John Steinbeck, who said, the journey is like marriage. So just to look a bit at what journey is. And the certain way to be wrong is to think you control it. And I think this is a very important part, right? We can go on a journey, you know, and that's what I want to do also with you, right? So I'm not controlling it too much. You come along, and we see. You don't necessarily have to understand everything, but it should be some flow and give you an idea of what incentivates myself. John Steinbeck is also important to me because I often use also kind of his famous book of Mice and Men. And my talk will be very much about mice and men. So you'll see that mice, of course, can be important models for humans, but you also have to bring this back to humans. A small little reference to Muybridge. We already heard, of course, about his life. And I find it actually, especially nice to actually receive an award after his name because, he was an imager. And I actually also, I think myself of an imager. My background is in electrical engineering, computer vision, and, basically I am imaging mechanics rather than maybe doing mechanics in that sense myself. And I think Muybridge was very much the same. He would not be able to put the formulas there why he was able to see famously to prove that a horse can fly, right? But I will try to use dynamic imaging today and to demonstrate to you that bones have ears. So similar to flying horses, I will show you that bones have ears. So I'm sure not everybody in this room is deeply familiar with bone. You all have them, that's good. And it's also important to know how dynamic bone is, of course. And this is just a nice picture here, of course, of showing a little bit of a femur and trabecular bone and cortical bone assessment here. And these bones get constantly remodeled, right? So every, let's say, 3 to 5 years, none of the pieces in your body in bone are the same anymore, right? You remodel that constantly, so it's totally remodeled. And these cells that are doing that are the osteoblasts and osteoclasts in terms of forming and resorbing bone. And in the middle of everything, we have these mechanosensitive cells, the osteocytes, that are believed to be orchestras of remodeling. So this remodeling interplay is illustrated in this graph here of normal remodeling, homeostatic remodeling, where you see bone is resorbed and then built back in. It's a sketch, that's not a simulation or something like that. It's just sketching this because it's not working quite like that, but similar to that. So bone remodeling allows actually the adaptation to new load conditions and also allows renewal of old bone material. So this is a very important process because, of course, as you are ambulating and sometimes exposing also your bones to certain overloads, you get cracks in your bone and they can be repaired, right, with this renewal. So you have a constant way of this material that is renewing itself. But the failure of remodeling leads to diseases such as osteoporosis and the associated fractures. And that's something that has been motivating me for most of my career, looking at age-related bone loss actually in that sense, and how can we look at that and how can we see what's going on there and why things are fracturing. And I'll show some of that in a second. Because one of the things that we see today is that biomedical imaging technology is really readily available, but very few attempts have been made to expand their capabilities by integrating quantitative analysis tools. So that, radiology is very, very big, right, in that sense. But the way to analyze that, you cannot find so much in a clinical settings, for example, in quantitative tools on these radiological pictures. So, but quantitative endpoints have become a very important factor for success, both in basic research and clinical practice. So, where the 20th century was a century for image formation, success in the 21st century will be judged by ability to extract quantitative information from images. I think I very much strongly believe that will be the case. And then from diagnostic and prognostic purposes, this microstructural imaging and strength assessment is really required for fracture risk prediction and treatment monitoring. And you can see that in some well-known pictures that from in the early 2000s, a lot of people saw, and you have these static images, of an osteoporotic bone in the spine of a human and a young normal. And if you have some training and looking at that, of course, you can imagine that they, not only have less bone mass, but also the mechanics is actually changing quite a bit because of that. But this is, you need to know something about that. But when you use dynamic imaging, like shown here, where we take biopsies and actually we just compress them, and every time we compress them for a certain step, we also take an image in high resolution. It doesn't take actually a lot, right, for you to recognize what's going on, what are the differences between a young healthy person and maybe an osteoporotic person on the other side. And you can see a couple of things that I want to point out. For example, that in the young normal, the fracture here is very much, defined by the process of the weakest link in the chain, where this is a little weaker up here, and that's where bone actually fails. Whereas in the osteoporotic case, this is failing everywhere, and that's actually not only a problem, to be able to predict where it's going to happen, but also for the biology that will come after that when you have a fracture healing process to take place, right? Because you have a lot of damage actually in that bone, that in osteoporotic bone, and the cells, limited sources of these cells, and they have to work everywhere to actually repair. So that's also a limitation actually, that this type of fractures are very dangerous because they also don't heal so well, because a lot of damage is going on, and somehow this will be distributed, the whole repair process. But nice thing about digital imaging is, of course, that you can also look at individual elements and where is actually happening. So you want to know where is this damage happening. And so you have lots of different elements. You can see there it's small little rods, and there's some plates in there, and we can look at each one of them and see what is the failure process actually in our simulations. So these are two of these examples here. And you can see typically if you have failure, some bending as well, and of buckling as well of these small little rods. But also plates can actually fracture. They are bigger elements, of course. They're stronger, stiffer overall, but they have these holes inside. And these holes are extremely dangerous, actually. So there's a typical process of the aging process that you actually start having these holes in the fenestration, we call it, in these plates. And if I go and zoom in a little bit in what's happening actually and use synchrotron radiation imaging, for example, I can actually see the cracks that are happening around that. And you can also see that these cracks will emanate actually from these convex surfaces and concavities that you have in that sense. And you can see that if you wouldn't have that hole, yes, you would have some cracks coming from the side, but many of the cracks actually emanate here. So that's why it's very dangerous. And it's really important, I say later on, in terms of counteract that to some extent. You need, of course, then treatment that allows you to fill in these holes. That would be very helpful, because as soon as you have filled in with very little change in the overall mass of your system, you have a much more robust system against cracking. So if you look a little even further resolution, so these are images that provided with 700 nanometer resolution in 3D. And now you can see actually all the cells inside, the osteocytes in the matrix. And if I take away the cells because they obscure a little bit my view, I can see that there is blood vessels inside, of course, this piece of cortical bone. And if you put this under loading, you can see that the cracks emanating actually directly from these blood vessels. So it's also important that these cracks, like any kind of void system that you have that is big enough to have enough energy produced to open a wake, then this is happening right there. And I think this is an important part, but also because these kind of canals are more numerous and bigger in the elderly population. So this explains a lot, right, why you actually have a failure there in that sense. And it's quite interesting, again, look at the resolution that you have, less than 1 micron actually in that sense. And you can see that how these cracks actually also go through osteocytic lacuna, right? And they actually emanated from this blood vessel right here, and then they actually go, the crack goes actually, the crack wake goes actually through these different little lacunas, and they play a big role in that. And we can actually use digital imaging correlation to look at displacements and strains in that. And you can see quite nicely, of course, that you have large displacements where the crack is, and it makes sense, of course, but you can see that early on already where it's going to heading towards because of the kind of front wave of this crack. And if you look at the strains, though, it's something else that is quite interesting and got us a good idea. You can also see that, these roundish structures, and they are associated with the osteocyte lacuna. And you can see that the osteocyte lacuna, they actually have higher strains around them, right? So they are intensifying these strains, and that actually is very helpful for them to be a good sensor. They're very well placed. They sense very early on that there will be a high loading. We then use computational prediction of these strengths. So this is a nonlinear supercomputer analysis. So it takes quite a bit of time on a normal computer, but in a supercomputer we can do that in only dozens of minutes, let's put it this way. And you can see, when you look at it already, you can see that it really nicely shows a very similar behavior than what we have actually also seen in the experiment. And of course, this gives us ways of having non-invasive analysis. So if you look at the nonlinear simulation, here that shows it's going to be damage accumulating actually in this plane. And if you look at the mechanical experiment, you will actually see that you have the same. If you zoom in a little bit, you see that even better. So I look at the simulation, and I would say these would fail these structures. And if I look at the experiment, I see it's exactly the same structures that have failed. So we have a way of doing a prediction of this experiment that is a more or less one-to-one behavior, right? So we can explain all of the behavior in this piece of bone with one fixed approach that is not, tuned in that sense, right? So it's a predictive way, and so that we can take this further then, of course, and doing such assessment in humans. In life, and we may be taking pictures today with such extreme CT, and they have resolutions of 60 micron in vivo in humans at the forearm or also at the wrist. At the tibia. And you can then, of course, do three-dimensional bone morphometry to compute the trabecular and cortical bone indices. You can use this so-called microfinite element analysis to assess bone strength. And we have done now also, some years ago, mechanical testing where we did have a lot of arms that were loaded in a testing machine to produce a wrist fracture that would be like a fall on the outstretched hand, and then the failure load was measured. So kind of a not so well-controlled test, but very realistic physiologically in how these will actually fracture. And you can see this is the type of image you can get if you take multiple sections. So this is not in vivo. These are from these cadavers. And you see that, of course, beautifully the internal structure. You also see that age doesn't tell you everything, of course. These are two male. And you can see the older one has actually a better structure than the younger one. This just to illustrate you where we have measured them, in that sense, very distal and towards proximal, and we checked, which are the most predictive regions actually for this behavior. And we can then, basically segment these structures, and you can see here, the three-dimensional structure that is coming out of that. Actually today we get even better resolution in vivo in real patients, and we're doing a lot of work right now actually to try to translate some of the knowledge that we also have from animal studies into these kinds of images. And then we can do this finite element analysis. And again, you can already see, the importance actually of load induction. You can see how the cortex gets loaded and also how these trabeculae up here get loaded. And it's by, very clear already that that's probably going to be a critical region, right? Because down here, you have much more uniform loading, and that's actually also the purpose, right? So this region must actually take up these loads and be able to translate it out to the cortex. So if we then do that for the many bones that we actually tested, you can see that you have a fairly good prediction of about 80% an R-squared value, and you need to have the most distal region actually to do that. So we learn where we should scan and what is a good predictor of this kind of behavior. So last part here on the in vivo strength assessment is just also, these are really in vivo data. And what we do here is to show you that this is the bone volume, bone mass, and this should be a very good proxy actually to, basically the strength that you have, right? You can see there's a high correlation between the strength computationally measured and the bone volume. But what I want to point out here, there is some 20 fractures and 20 controls in this study here. And if you look at that, you can see, for example, that this is not just error of the measurement. These is real change. Here you have the same strength in all of these samples, whereas the volume actually changed quite a bit, right? So this is not, if you just measure volume, you would have thought that this one is much weaker than this one on this side, but that's not necessarily the case. Even worse are cases like this, where you actually have the same volume. From this, clinically, you would predict that they would have the same strength, but you can see that there is some 50, 60% higher strength actually in those two samples versus these two samples here. And then it's interesting to know actually that those are the ones that actually fractured, and these are the controls. right? So it tells you already that if you just look at the classical clinical measures, like DEXA, for example, that pretty much just measures mass, that you're not very well to interpret why, basically the failing or who should actually fail. So a strength measurement is actually helping you. And the nice thing about these strength measurements is they can also, of course, create from these type of images, you can already see some differences between the shape of the two parts, the ones that actually failed, with the same overall mass than this one. You can see these are larger areas to some extent, but smaller cortex. And if I do the finite element analysis, you can also see that here the cortex is relatively protected in the ones that were controlled with the same mass, whereas here is very thin cortices, and this of course gets lots of strain in there in the critical area where you start having failure. So just to have the density by itself doesn't actually help you so much. You really need to see the full picture, right? And the last part here that I would like to talk about from the in vivo side is that if you do a nonlinear analysis and you look at the plastic behavior of the material, you can see that you get a lot of plastic deformations actually right at this interface, and there might also be a little bit of problem of the boundary conditions. But if you look in the cases, you see a pattern that looks like that, a lot of plastic deformation actually everywhere in the cortex. And again, you have the same problem that you have now many, many weak points, right? And they will actually start failing, and that's where we see the whole action going on. And we can learn from that, and we see that if we use such approaches and use plastic failure or plastic deformation and plastic strain in our cases, and then we actually have 50% better prediction to disassociate controls and the people that actually have a fracture. So with all of that, of course, this has been a lot of that work has been done over the last 20 years. But of course, you start also thinking about, once you know these mechanisms, can we actually stop that, right? Can we stop osteoporosis? Can we prevent fractures? And probably all of you know, of course, we have effective treatments today. So these treatments do help you to reduce fracture rate by about 50%, but they have side effects, these pharmaceutical interventions, and these side effects, they are quite rare. But people would not want that, right? So they could lead to atypical femur fractures. They could have necrosis of the jaw. Very few people are affected, but if you have that, then it's not very nice. And that's why people shy away, actually, from getting the therapy. So today we have a certain crisis, basically, that the drugs are available, but the elderly population is not willing to take them. And so you should think about maybe other alternative ways of having treatment that would be, more hopefully less side effects, although it's always difficult, of course, to have treatments without. So, and one of the things that we are interested, of course, is to use mechanical forces to help the bone build and or maintain. And this has, of course, been all laid out already by Wolff. So we work a lot with Wolff's law, and Julius Wolff stipulated in 1882 already that bone forms and gets stronger when it's loaded. Is bone not or only lowly loaded, it will resorb and get weaker. So he observed that and looked at a lot of mechanical interactions, actually also with Kuhlmann, who was calculating cranes at ETH Zurich. And so there's an early connection actually to ETH also of this kind of work and from Wolff. And tennis players are a good example, right? So we all know, or baseball. Pitchers, for example, is another one that they would have in their playing arm a lot higher bone mass than in the non-playing arm, and this has nothing to do with if they actually do that with both hands or just with one hand, because they always serve only with one hand. So it's the serve that actually plays the biggest role. It's not the playing during the game, right? Because especially Federer, he only serves more or less, right? So, and he doesn't want to run after that, and so that's that's why he gets it, right? So and that's quite interesting. Other people that have not a lot of problem actually with creating great bone mass are bodybuilders, and they have maybe other problems, but they don't have bone problems. And on the other side of the spectrum, you have the astronauts. And of course, with the weightlessness, they actually lose bone, right? And it's a big problem as well, because they lose about 2% bone mass per month. And of course, if, for example, if they're training to go to Mars, which is more or less like 4 years or something, or 3 years of traveling, they will not come back with any skeleton if you don't protect them. So it's very difficult to actually do that, but pharmaceutical interventions are also something that they're of course thinking about, and that's a certain concern, is a big concern actually. So vibration therapy might be a new perspective to build stronger bones. This has had a lot of scientific studies to show that bone is very susceptible to mechanical vibrations. And vibration therapy is currently investigating as a potential therapy against osteoporosis. There is still quite a bit work needed to understand exactly, how you can scale from small animal work, where we know exactly that this is working really well, to the human scale. But this kind of exercise on plates is something that the people want to do, of course. And this vibration is basically sound, and that's where it comes from, that bones must have ears, because the bones, they need to hear that, actually, that sound, right? And we are interested in how do they hear that? What is the mechanism actually down to the cellular and molecular scale? And that's what I like to show you a little bit, how we approach this technologically. So what do we know so far? So the maintenance and adaptation of bone morphology results from this orchestrated remodeling process. And this is locally coordinated by osteocytes with biochemical signals, of course, resulting in increased or decreased bone formation and resorption activities. And so to better understand bone adaptation, we therefore have to understand how osteocytes sense, process mechanical loading within their local microenvironment. So really locally, not as an ensemble, but individually. And I'll show you that this is actually quite important. So here is where the mouse comes in. We use a mouse model to do that, where is a mouse model of bone adaptation, which is using vibration therapy. And so we are actually looking at the caudal vertebra in the mouse, and so at caudal vertebra 6, and we push pins in adjacent vertebra, and then we can use cyclic loading with 3,000 cycles and 10 hertz and 8 newtons. We do that 3 times a week, and we do that for 4 weeks. And then we use such CT scanners, micro CT scanners, that allow you to put the animal in, and they work like the clinical CT scanners, so they're not like the normal desktop CT scanners where you have a probe that is rotating. You have now the rotating tube and the rotating detector, and they can provide you resolutions about 10 microns in vivo, so extremely fine resolution, right? And what you see is something like that. So just to also show, this is now over time, over 4 weeks, how the bone is changing because of mechanical adaptation. And you can see you start with this, and then you have this thickening process actually going on when you do the mechanical intervention, when you do the vibration therapy. And if you look at the quantification of that, you can see that in this 4 weeks, with just 3 times a week, so 12 training intervals, let's say, right? You get about 20% more bone in these animals. So it's very potent as an anabolic drug to actually do that. So I want to quickly go with you to visualize this load adaptation process at the even high resolution, to also show you the fidelity, right? That this is, remember that this is done in vivo, and these structures are on the order of maybe some 20 to 40 microns in that sense, right? And I'll just go, this is baseline, and after 1 week, we already see some effects, right? That some of the structures have been enforced, and actually, you will also see that, and I talked about that earlier, these holes, right, that are quite dangerous, they start closing, right? You see that here after 2 weeks, after 3 weeks, even more close, you get thicker elements, after 4 weeks, even further on, right? So it becomes less of a liability, let's say, and eventually these holes can actually close. So what happens here, what I've shown here, is because we have now multiple timed image, and where we see that we basically can look at where bone is now formed and where bone has been resorbed, because we have multiple images. We subtract these images from each other, and every time we see a voxel in a new image, then we know that wasn't there in the old one. We know that bone has been formed, and wherever we see something that goes away, right, the voxel disappearing, bone has been resorbed. And so in orange, you see all the bone that has been formed, and it's also interesting to see when you have this anabolic response, you're not having everywhere forming surfaces, you also, of course, have some resorption going on, and that's what's shown here in blue. So this is also interesting question, how is this actually, controlled. And this is something that is important to see. Then you have this very important structure here that gets thickened. And because of that, most of the load will actually go through this side here and will unload this region. And then the theory of Wolff would say this should be resorbed. And it seems something like that is happening. And I'll show you that a little bit more, how can we actually do that? And so this is the real flight through bone and some journey with you. So we're going into a arterial support, actually, nutrient vessel into the bone inside. And what we've done with this bone, we have actually used finite element analysis to load it, right, virtually. And when you look inside here, you will find areas where bone now is highly loaded. So this is indicated in red. And other areas where bone is, almost unloaded, right? And Wolff's law would now tell you that those areas that are unloaded, like this one, they should be resorbed. And the areas where you see high loads, like here and like here, there should be more bone formed. So because we have now these different maps, we have the formation and resorption map that I show you here, and we have the map initially, how was the loading actually, and we see that, okay, some certain structures are overloaded, and yes, in these structures, we actually form bone, and some structures were underloaded, and yes, some of that was resorbed over time. And you can look now at each of these places and quantify that, and you can show statistically that this is actually the case, so that you have more bone forming where you have high strains, and bone resorbing where you have low strains, compared to the places where no bone changes are happening. This we can show that in all applications, not only in the loaded animals, but also in the control animals this is happening. And all kinds of therapies we have done with the animals, this is always the case. So this is a kind of a universal law that whatever happens, bone is actually able, also in old age, to do it the right way. So it's always under this control. And that's quite fascinating that this is something that is to about 60%, we can say that everything that is happening in the mouse is actually guided by mechanical forces. So this is a very important part. And then, of course, we are interested to look at how these then go towards the cellular aspects. How do the cells feel that, and what do they do, and how do they communicate that? So we will do the organ scale loading. This results in a strain distribution. And then on the cellular scale, we have the osteocytes that feel high loads, and that would actually attract with their signaling osteoblasts to form more bone on these surfaces. And when you have low loads, that should actually attract osteoclasts to resorb. And then we can look at it, of course, at genomic, transcriptomic, at the proteomic level, and for each individual single cell, and see how does this actually contribute to the overall picture. And this will then lead to a coordinated remodeling effect that then leads to a new map of bone adaptation. And we would like to actually use this approach, and we call this a local in vivo environment imaging approach, where we have in vivo micro-CT, and then we have a second later time point that we measure. We basically match these with each other and can see where the bone remodeling is happening. But also we can do the mechanical strain energy density measurements. And if I now, at the end, take a histological section, right? So this is now no longer in the live animal, of course, but this has been measured in vivo, and this we take at the end point of it. And we can do more now, right? Because in these sections, we can see the biology, and we see the cells. So we can look into a small area, like this one, and we see all of these osteocytes inside. And then we have a technique using laser capture microdissection to cut out each cell individually. And I'll show you that just in this video here. So what happens is that you don't see well the cells in here, but you can believe me that underneath here would be a cell. And you take a laser, and then basically you go 3 times around it until you have taken this out of basically almost, freed up this space, and then you use a laser pulse to eject actually that piece of material with the cell inside. And then you do that for hundreds of cells, right? And you only have half an hour time, because otherwise all the RNA is degraded in this part, but you have to be so quite quickly. But then we have
individual cell material that carries RNA and all kinds of things, of course, inside there that we want, also proteomic information. And we have not only each cell, but also we know exactly the position of these cells in the in vivo environment. So we can link it up to whether this was an area that got resorbed, close to a resorption area, or in a formation area. We also can check whether this was medium loaded or highly loaded or unloaded. And so we can make basically the connection between each cell and how was their life, right? How was the history of the cell? What did it see in the last 4 weeks, right? And that's actually an important part. Then we can do a biochemical analysis of each cell or also groups of cells. And we can do qPCR, or actually we started doing RNA sequencing of these kind of singular components. So we have a lot of information about the genetics of these cells. And I bring you an example how we map gene expression in these single osteocytes. It's not so important what these genes are. This is SOS and OPG and rank ligand. For the ones that are in the field, they will recognize that. What's important, though, is that this rank ligand signaling is there for attracting osteoclasts. So it's something that will form resorption. And you can see that if you look in the control animals, you see the rank ligand is actually not detectable when you have very high loading, which makes sense, right? When you have very high loading, then you should not have any osteoclastic resorption at that point. So that gene is downregulated, and you see this is a logarithmic scale. And you see in the medium range, this is 10 times lower than in the low range. When you have very low loading, then there's lots of this signal available, whereas some of the other parameters are not so strongly regulated. You can see that SOS gets downregulated. You would also expect that because this is something that is actually ubiquitously expressed typically, but with mechanical loading should be downregulated. And you can see that, of course, at local sites, it is downregulated a little bit. What happens if you have a loaded animal? You can see SOS overall is downregulated, but not so specifically in high and low loading areas. But with the rank ligand now, you actually cannot even detect it on the medium level. So this extra loading bout that we give them protects them from osteoclastic resorption. So very important. So it's not necessarily anabolic in that sense that it produces a lot more bone, but a lot less bone is resorbed, right? This is what this mechanism is behind, and that's quite important. We can look at, so for example, the SOS signaling a little more carefully also using immunohistochemistry, and I show you some examples there. So all of these cells are stained, and you can see that sclerostin is in brown. So that, if you have a cell that has sclerostin expressed, then it's brownish, and the bone matrix is in blue. And, and of course, with no loading, you have a lot more sclerostin, as you would expect. Then with 8 Newton loading, this is kind of downregulated. And now when we look at, can calculate, we can look at osteocytes that came from areas that are underneath a resorbing surface in a quiescent area or an area where there was bone formation happening. And you can see that the sclerostin expression is downregulated significantly compared to both the quiescent area and the resorption area when you have forming cells, right? So this is an important finding in that sense. But what's more striking even, I found, is that when you look at it and look slightly different, you look at the dynamic morphometry and you look at mineral resorption rates and mineral apposition rates, right? So how fast is bone resorbing and how fast bone is actually forming. And we look at high levels and low levels and, basically zero formation, resorption, and then the other way around on this side. And if I plot this, and you look at the sclerostin expression with this method, you can see that you have this very high control, right? So that 96% correlation, 84% correlation in another animal, and it's very important, right, to show this. And what it also shows you, though, if I would have just plotted this for the 2 animals in one picture, not for each animal individually, I would not actually have seen that they have this high correlation, right? Because if you plot that all for different animals, they're not exactly on the same slope. They're also not exactly, at the same offset in that sense. So that's quite important that this technique allows you to look individually in each animal because each animal is kind of its own control, and that's quite important. So you can also save animals, and it's important to do that. You have the same mechanism happening in the animals, but you don't actually need to go for many, many animals to do that. So lastly, I'd like to show some images here from live imaging that have to do when you go from 2D to 3D, because histology, you can also do histological three-dimensional sectioning. And when you look at what's happening here, sclerostin, normal sclerostin is in black, and sclerostin downregulated cells are in red. And you can see in terms of when you look at just 2D, you don't notice actually a lot of patterns here, right? It just seems to be random. With SCD, well, they should be downregulated when you have high loading. Yes, this might be the case for those reds here, but not so much up here. But if you go in 3D and look at these parts, then of course you see actually sometimes much better that, for example, these cells that get downregulated, they're close to formation. We said that this actually downregulates it, right? In 3D, you see that much better. Here, you think like, well, there is no formation actually going on here. But in 3D, you see these cells are very close to this surface. So it helps you a lot to actually work directly in 3D and not just in a two-dimensional fashion. If one thing that's also quite important is that these increases and decreases over time are driving it. So you need the history, right? So if you look at the changes in SCD over the weeks, this is also helping you a lot to understand, you know, that if you have an increase or a decrease of SCD over time, you actually have a change in this protein expression of sclerostin, which is the protein that is coded from SOST. So it just gives you an illustration and gives you the last picture here of a vision that we have, that we want to do, basically be able to do formation resorption maps. We want to look at the loading in vivo over time of these animals. And then we can actually do histology in 3D after the end, and we're going to see each cell in the structure actually, and we know exactly, you know, is it associated with high loading? Is it associated with resorption or formation? And this will give us a wealth of information. So on very small numbers, so because you can look at each area is its own, again, control. I think this is an important part. So this is a little bit more like a vision, because this is very time-consuming to actually do that, right? So this is a technique, not yet a technology. But my belief is that with enough efforts on our sides and hopefully others in the field, that we can get that to be a technology that is basically turnkey, and you can do that, you know, all the time and in many, many labs around the world. So in conclusion, I would like to say that bones have big ears, and they have very good hearing. And it's important to know that hierarchical investigations of bone function facilitates better understanding of osteoporotic fractures and hopefully their prevention. The time-lapse in vivo imaging allows longitudinal quantification of bone remodeling and adaptation using dynamic morphometry. And mechanomics allows coupling of biochemical information with the mechanical and remodeling microenvironment of in the cell vicinity. In the future, this will facilitate better understanding of biochemical signaling and load-induced bone adaptation, and hopefully new treatment options against osteoporosis and osteoporotic fractures. So I strongly believe that with this understanding, we can do something there that use also a treatment that is maybe more physiological rather than, you know, chemical in that sense. So I think this is my hope, and working towards that goal, of course. So with this, I'd like to stop, but not before also thanking, at least the visual picture, the many people that actually made this, I think, a success. And so just go through some impressions of coming from 2004, a relatively small lab, to building up an institute together with other professors at ETH Zurich, where we now about 150 researchers, and becoming also department head and having 500 researchers with time, to now in the newest days, no longer any duties in the department, having my small team again, and playing around with them. And with the help of our funding agencies, we can actually, I think, do very good things for the years to come. And with that, I thank all of those people, but also thank you for your attention. And this is the end of it. Thank you very much.